லைவ்ல தானே இருக்குப்பா. அந்த தள்ளி வெளியே போடுங்க. பட்டன்லேருந்து பட்டனில் படம். இந்த சைடு இது ஒரு நடுவில் இருக்குல்ல. இந்தாங்க, இது இருக்கு.
இதை இதுக்கு முன்னாடி வைக்கலாம்ல?
இதுக்கு முன்னாடி
இந்த ஃபோனை இதுக்கு முன்னாடி
இல்லை இப்படி தான் இருந்துச்சு.
சரி,
சேஃபாக இருக்கும்.
நிற்கிதா இல்லை இன்னொன்று போடுவோம்?
இன்னொன்று போடுங்க.
இன்னொன்று போட்டு
சேஃபாக சேஃப் போட்டுருங்க. நிற்கிது. சேஃபாக சேஃப் ஒரு நிமிஷம் போடுங்க. இருக்கா ப்ரோச்சர்?
ஓகே. இது ஒரு ஆள் இங்கே இருக்கிறவங்க சொல்லிட்டு பார்த்துட்டு வரேன். யாரும் பார்க்குற மாதிரி இருக்குது.
கிராஸாக இருக்குது, ஒன்றும் இருக்குது. இது எடுத்துடலாமுங்க. இருக்கவா
இந்த இடத்துல வைக்கலாமா உள்ள? இதை கழட்டி எடுத்துடலாம் இல்லையா?
இது கழட்டினா நிற்குமா?
ட்ரைபாடே தூக்குதுராடி. டேய் டேய், அது வச்சாதான் கரெக்டாக இருக்கு. வச்சிடறேன்.
ஃபோக்கஸ் ஆகுதா? பட்டன் மட்டும் மென்டல் நோட்டி.
பட்டன்
உள்ளே வைக்காதானே சவுண்டு?
இந்த கேமரா பின்னாடி
கேமரா தான். ஆமாம்.
இல்லை ஓகே, இதில் தெரியல. முன்னாடி தெரிய வச்சுருவோம்.
முன்னாடியா? தள்ளி தள்ளி. முன்னாடி தள்ளி
லைட் ஆஃப் பண்ணிட்டா
சும்மா நீங்கள் பாருங்க. ஓ இதெல்லாம் ஆன்லைனா? ஓகே. இது ஆன்லைன் ப்ரெசன்டேஷனா? சரி. சாருக்கு மைக் கொடுத்துருங்க.
Students are requested to return to their respective places as we continue with the program. Kindly take your seats, everyone, as we are about to proceed with the next session. yoga, all the students are requested to take their seats.
Questions are questions getting questions.
இல்லை அது வழக்கமாக நடக்கிறது.
The university lots of practice.
Poster presentation and model presentation participants are requested to assemble near their
This
displays.
is explaining you 5%.
ஓகே
It can be professional practice.
ஓ
I repeat, poster presentation and model participants are requested to assemble near their displays.
Up to the university. The sir is invited to professional practice.
ஓகே
Oral presentation participants are kindly requested to assemble near the podium.
Even online monthly once
ஓகே
the qualification
ஓகே ஓகே
ஓகே நியூ எஜுகேஷன் பாருங்க லைசன்ஸ்க்கு பிஎஸ்சி ப்ரொஃபஸருக்கு ப்ரெசன்ஸ்
ஓகே
இண்டஸ்ட்ரி பர்சன் நிறைய கொடுப்பாங்க அது ரீசன் என்னன்னா இது வைஸ் சான்சலர் இந்தியாவில் அவங்க வந்து இதுவே காசு எடுக்கிறாங்க
ஓகே
இண்டஸ்ட்ரி பர்சனுக்கு வைஸ் சான்சலர் போடுறாங்க காசு உள்ள போடுவாங்க
கரெக்ட்
அவங்க இருக்குன்னு அவசியம் இல்லை பணம்
பிஸ்னஸாக இல்லாமல் ஒரு
அவங்க சொன்னா நான் ஸோ கான்செப்ட் வேறு மாதிரி என்னன்னா அகாடமிக் இருக்காது அது சொல்லக்கூடாது எதுக்காக நம்ம வந்து படிக்காத கூட எலெக்ட் பண்ணுறோம் காமராஜர் படிக்கல இல்லை இல்லைன்னா நேர்மை மட்டும்தான் இருக்கும் என்ன இருந்தால் போதும் தெரியலன்னா இது என்ன செய்யலாம் கேட்டுட்டு போகலாம் அந்த டீச்சர் இருக்கும் அந்த கான்செப்ட் டீச்சர் வந்து சொல்லிக்கிறதுக்கு எனக்கு தெரியல ஐஏஎஸ் ஆஃபீஸர் போகிறாங்க இங்கே இண்டஸ்ட்ரி எல்லா ப்ரொஃபஸர் இருக்க போகிறாங்க இப்படி செல்லாம் இப்படி செல்லாம் இப்படி செல்லாம் எது கரெக்டான டீச்சர் எடுக்கணும் அதுக்கு மட்டும் தான் ரைட் எக்ஸாம்ஸில் வந்து இங்கே வந்து டே டு டே ஆக்டிவ் ஃபாரினில் அப்படி கிடையாது அவங்க ஃபண்டு கொண்டு வரது தான் வேலை
ஓகே
இங்கே வந்து எல்லாமே கல்வி கொடுப்பாங்க லீவ் கட்டி கல்வி கொடுப்பாங்க
Poster presentation and model presentation participants are requested to assemble near their displays. Oral presentation participants are requested to assemble near the podium.
யூஜிசியில் போர்ட்டல் ஒன்று இருக்குது
ஓகே
போஸ்டரை ப்ராக்டிஸ் யூஜிசி பண்ணினா போதும் போஸ்டரை ப்ராக்டிஸ்
ப்ராக்டிஸ்
Oral presentation participants kindly come forward.
ஓகே எதிர்காலங்களுக்கு நாங்கள் சொல்லணும் ஏன்னா நாங்கள் இன்னொரு இன்கம்
இல்லை எனக்கு இன்கம் இதெல்லாம் சொல்லி பண்ணுங்க நீங்கள் பார்த்து என்ன சொன்னீங்க ஒன்லி ட்ராவல் எக்ஸ்பென்ஸ் பண்ணுங்க நீங்கள் இது கிடையாது ஆன்லைன் கிடையாது ட்ராவல் எக்ஸ்பென்ஸ் மேட்ச் பண்ண போதும் மற்ற காலத்துக்கு வரலாம் ஆன்லைனில் வந்துக்கலாம் மந்த்லி ஒன்ஸ் வரும்
ஓகே
உங்களுக்கு கன்வீனியன்ட் உங்கள் வந்து நீங்கள் இப்போ நீங்கள் ஒன்றே வரையும் வச்சுக்கோங்க ஹோல் டே 8 hours class
Please be ready for the oral presentation. Ms. Praveshya, please be ready for the oral presentation.
Praveshya, are you ready?
Yes, ma'am.
இல்லை அவங்க சவுண்டு சத்தமாக இருக்கும்
Kindly wait for 2 minutes.
பிபிடியா
As curated, participants will be terms of 2 or 3. Each presentation is allotted a total of 10 minutes, including 7 minutes of presentation followed by 3 minutes of discussion. Presenters are expected to communicate their ideas clearly, manage time efficiently, and ensure a well-structured delivery for oral presentation. Thank you. We request everyone to kindly settle back.
ஓகே
அவங்க ரெனுமரேஷன் அப்டேட் பண்ணாங்கன்னா ரெனுமரேஷன் வேணால் டிக்ளேர் பண்ணிக்கலாம் அவங்க ஒரு எக்ஸ்பீரியன்ஸ் இருக்குது நீங்கள் என்ன சொன்னால் எவ்ரி 3 இயர்ஸ் மார்க் ஆகிடும்
ஓகே
எவ்ரி ஒரு 2 to 3 இயர்ஸ் தான் ஒரு யூனிவர்சிட்டி
ஓகே
அதனால அதனால் நீங்கள் எவ்ரி 3 இயர்ஸ் நீங்கள் மார்க் ஆகிடும் லிங்க் பண்ணுவாங்க
Kind attention. For poster presentation, all posters must be designed in a portrait format. Participants are expected to ensure that their posters are neatly displayed. For model presentation, participants must present at a working static or semi-functional model that clearly demonstrates the concept with proper labeling and explanation.
Present பண்ணுவோம். ஸ்டார்ட் பண்ணலாமா? ரமேஷ்.
Let us now begin the session. Parishya, please start. Can I start now, ma'am?
Praveshya, now you can start.
You can start now.
Kindly share your PPT. Share your screen.
Is my PPT visible, ma'am?
Yeah.
Yeah, ma'am, visible. Good morning to everyone. I am Praveshya from Army Engineering College, Kumaran. I am here to present about creating relevant orthotopic mutants.
மியூட் பண்ணிவிடுங்க
Orthotopic mutants means they are microorganisms that are not able to produce their essential nutrient. Here what we do is to create orthotopic mutants of glutamine. My problem statement is that to create antibiotic resistance, to create an orthotopic selection system instead of antibiotic resistance markers, as they are high of cost and have so many disadvantages. The absence of well-established orthotopic strain restricts the development of a marker-free constitutive expression platforms in isotopic. Here what we are going to do is to create an globally orthographic mutant and complement it in isocyclium so that we can enhance the DHA content in it, which is an omega-3 fatty acid. My aim is to develop antibiotic-free glutamine oxidotrophic selection system in isocyclium by generating glutamine oxidotrophic mutants and escaping them via glutamine synthase. We actually develop oxidotrophic mutant and then we indulge glutamine synthase gene, complement it, and then transfect in isocycliums to enhance their DHA levels instead of using an antibiotic resistance marker. Where glutamine is produced by PCA cycle, alpha-ketoglutarate, glutamate to glutamine by glutamine synthetase gene, which we are going to knock out here. And our work here is to first select a strain, a isocyclium strain, which can perform efficiently in our growth conditions and optimize media for strain to grow and start UV mutation. We produce oxidotrophy by UV mutation, but we had a little problem with it. We shift our strategy to glufoxamine, which is a metabolic inhibition pathway, where we select the surviving cells and screen the oxidotrophy, confirm the phenotype, and confirm the oxidotrophy by taking in glutamine and glutamine present supplemented media. Strain selection. We actually selected 101 strain here. It's efficient and can produce in high quantities. We selected 101 and 108, which had better growth. Here we used 1011 strain, and these are the cytocyclic cells which we viewed under fluorescent microscope. The optimized media we used here is ASW media with a little bit changes. We call it as minimum media, where there are no krypton, yeast extract and other components, only salt concentrations. We differed ammonium sulfate and potassium nitrate concentrations from 1 to 10 ammonium sulfate and potassium nitrate from 7 to 10, which we got 10 grams of the sandage concentration for ammonium sulfate and 7 grams for potassium nitrate. We took OD measurements for the growth to be checked and to optimize the media, where we had this OD values of the peak graphs. And we check for growth and viability assessment. Viability we check using one side where the same cells are dead and the other are alive. We checked it with fluorescent microscope and we counted the cells with hemocytometer. By 1/10 dilution, we used an orbit and take 10 million cells and we start with UV mutation. For UV mutation, master gauge was cut with M1 agar and the screening was done with minimum media with glutamine and glutamine. Mutation, we prepared the culture and took 10 million cells. We washed it with PDS buffer and then we gave it for exposure within 10 centimeter distance from UV, 254 nanometer for 30 seconds, and we kept for dark incubation for 15 minutes and we recovered the cells and created in M1 agar with glutamine. We carried out more than 10 trials. These are the mutant plates. And we collect the single colonies and plate it in massive plates. We have about 534 colonies from our mutation. And master plate colonies were screened in glutamine present and glutamine free media for its growth checking. These are the colonies that we plated for screening. And as they were same results in both the plates, we went for glufosinate treatment, which is a metabolic inhibition process where this glufosinate will inhibit the glutamine metabolic pathway and knock out its gene, glutamine synthase gene, so that an oxidotrophic mutant will get created. Now we started with sensitivity assay to know which concentration is best to knock out this glutamine synthase gene and when the growth is able to stop. And we plated in 5, 10, 15, 20 concentrations. Now after this, we know which concentration will knock out the glutamine synthase gene, and then the oxidotrophic mutants will get collected and will be screened in determined percent and free media, where the observers are selected and complemented with determined synthesized gene. And in size of item, the DHA levels will be checked later. Future goals: there will be molecular characterization of mutants, heterologous protein expression, development of stable oxytocin selection system without antibiotic resistance markers, genetic complementation studies, and metabolic enzyme applications. Thank you. Any questions? Thank you, sir. If any doubt, please ask me, sir.
Hello. Can you hear me? Hello.
Yes, sir.
What is the purpose of your choosing the glutamine out of the 20 amino acids?
Sir, cytovitamins are microalgae, marine microalgae. It does rich DHA content, but we are using antibiotic resistance markers like xeosid, which are very costly. So we change our strategy. An oxytocin selection system will be used so that antibiotics can be rejected and this can be used instead of that. It will be of reduced cost, no other disadvantages. It will be very beneficial for us.
Is it your BTech student or scholar, PhD scholar?
BTech biotechnology, sir.
Okay, final year.
Thank you, sir.
How long you took this project to complete so far?
3 months, sir.
Okay, thank you.
Okay, Mark. Thank you.
Thank you, sir.
Now I invite Sri to start their presentation. Kriti, can you hear me? Yes. Yes, Kriti, you can start now. Yes, Kriti, you can start now.
Good morning, everyone. I'm Priya from Canada Engineering Science, Tirunelveli. I'm doing my final project in Biotech Life Sciences project. My title is Enhancing Tumor Biosynthesis in Cytometry. The problem statement is increasing demand for steroid in pharmaceutical production, limiting limitations of animal-based systems and synthetic steroid sources, high cost and sustainability issues with synthetic production, no steroid is due to competing metabolic pathways, need for an easy, scalable microbial production. So, usually, cytovegetable is a microalgae which produces steroids in high content, but everybody generally don't use animal-based systems. Typically don't prefer animal-based to an alternative to prepare a plant-based vegan steroid source so that everybody can be aware of this steroid components and other pharmaceuticals. So steroids are lipid molecules that are naturally present in plants, animals and monkeys. These are all highly demanded in pharmaceuticals. Cytovegetable and malleable unicellular heterotrophic microorganisms. These are naturally with high content producing lipids and steroids. My aim is to explore the potential of cyclocytidine as a microbial host for enhancing metabolic pathways through controlled cultivation and metabolic engineering. The objectives are to study steroid production in cyclocytidine, to select a suitable strain for enhanced steroid accumulation, to optimize metabolic engineering protocols and cultivation conditions for improved growth and steroid yield, to analyze and compare steroid content under different conditions. So this is my work plan. I initially selected strain and then using UV mutagenesis, then cultivated the engineered strain and then extracted the steroid content and analyzed with PLC and HPLC. So these are the pathways involved in this steroid production, and this screening and screening to steroid pathway is this way the steroid is finally producing. Selection of strain. I used wild type cyclocytidine strain of 1011, which produces high steroid than other strains. It requires pH of 6.5 to 7.5 and temperature 25 to 30 degrees Celsius. Its time is usually 24 to 48 hours. The media required is M1 media, which follows the composition as follows: potassium chloride, calcium chloride, magnesium chloride, sodium sulfate, yeast, peptone, and dextrose. These media are autoclaved using this autoclave. It is known for its sterilization. The original strain is subcultured to the media and then incubated overnight at 28 degrees without continuous shaking, and the fully grown cyclocytidine cultivated. Using fluorescent microscopy, the growth and contamination is checked in 40x focus, and the viability is checked using event size. Cell counting is done with cytometer, where the average cells is counted and its dilution is considered for its total cell count. So usually we have plated 6 plates of different steroid level and grown that cyclocytidine in every plate to check at which concentration the microbe is having higher growth. In this, we chose 0, 100 mg, 250 mg, 500 mg, 750 mg, and 1 mg of steroid content. We selected this 250 steroid plate, which shows a good growth than other plates. So we standardize and optimize this 250 mg for this cyclocytidine growth after UV mutation. So for this UV mutation protocol, we have initially collected 10 million cells in a bin of 22 Swiss and followed by previous wash and sterile wash. We transfer it to petri plates with dilution of the cells with water and expose it to such UV light for 30 seconds at 20 to 54 nanometer wavelength. After this 30 seconds, we are given 15 minutes of dark incubation, which shows the mutation. This is the mutation setup for this cyclocytidine. After mutation, these cells are collected and plated in one agar plates, and we kept it for 48 hours for actual growth of mutation plates. Then we have collected the single colonies and then cascade with cyclocytidine, and it is considered as primary screening of colonies. These are all the mutation plates we did. We did some 10 trials, out of which 26 has good growth. Finally, we did master plate, which has around 434 colonies of UV mutants. This is called an primary screening. And this primary screening is done with 0, 250 mg, 750 mg, 1 mg to know at which level the mutation is occurred, and we also quickly colony at 1 mg to know whether the wild type is mutated or not. So out of this 434 colonies, I selected 50 high yield colonies and 50 low yield colonies and plated it in 0, 750 and 1 mg plate. This is showing and out of these 100 colonies, I chose 5 high-yield colonies and 5 low-yield colonies to quickly decide. And these and that's it. I'll do this really activity population and experience. Thank you. Any questions?
Hello. Hello, can you hear me? Hello. Hello.
Yes, ma'am.
Yeah. What is the application of this mainly? Whether you are focusing for the industrial application or other business application?
Sir, I don't hear you, sir.
What is the application your project?
Usually, steroids are produced from animal-based sources.
Okay.
So I am choosing an algae which can be prepared by everyone, and everybody can avail of this steroid source instead of depending on animal source only.
Okay. So whether you conducted any test like animal studies, only you did some in vitro studies, some of the in vitro studies, right? Whether you conducted any animal studies?
So the voice is not audible, so it is echoing.
Hello. You conducted in vitro studies. Either you plan for in vitro studies in the future. Hello.
Sir, can you repeat it, sir? Can you repeat it?
Yeah, you conducted in vitro studies. No, you have shown some slides in the in vitro studies. Okay, ma. Bye. Good.
Hello. Is it my voice is clear?
Yes, sir. Yes, sir.
Your presentation is excellent. If it's you have a slide number and content of presentation and summary would have been better. I mean, otherwise the presentation is excellent. I have a few questions. First thing, you did mutation through UV mutation, is it right?
Yes, sir.
In UV chamber, in laminar flow.
Yes, sir. Yes, sir.
Okay, great. So, did you able to analyze genetic pattern also?
So we are just doing random analysis and taking it to different concentrations, and finally we are selecting the colonies out of only screening methods. We don't involve any genetic level analysis now.
Okay, great, super. So, the project is excellent and the documentation is excellent. Thank you.
Okay, sir. Thank you, sir.
Okay, ma. And the best. Bye.
Call upon Kadaksha to present.
Yes, ma'am. Am I audible?
Yes. As a reminder, please try to conclude within 7 minutes.
Yeah, okay, ma'am. I hope my slide is visible.
Yes, visible.
Shall I start my presentation, ma'am?
You can start now.
Hello.
You can start now.
Good morning, everyone. Myself Kadaksha from Mahatma Gandhi Medical College and Research Institute, Pondicherry. I am pursuing my final year MBBS. Today, I will be presenting an emerging concept in preoperative care, that is, programming the analgesic microbiome. This is a preoperative strategy to reduce pain. Why it is needed? This is because surgical or operative pain remains a major problem despite anesthesia.
High opioid use also causes many complications like respiratory depression, nausea, and dependency. This shows the need for safer non-pharmacological strategies. Recent research have shown that gut microbiome influences pain pathway. What is it? This approach focuses mainly on modulating the gut bacteria to influence the pain pathway and improve the surgical outcome. This is based on a concept called gut-brain axis. It is a bidirectional communication between the gut and the brain. It involves neural, immune and hormonal pathways and also involves vagus nerve signaling. The microbiota plays a very important major regulatory role. The microbiome affects pain through multiple mechanisms. The microbial metabolites has its direct influence on the nerve activity. They will alter the cytokine production, affecting the inflammation, and influences the central pain processing and neuroimmune signaling. I'll first talk about Akkermansia ministerialia. It's a beneficial gut bacterium. It enhances intestinal barrier, reduces the systemic inflammation, and decreases pro-inflammatory cytokines like interleukin-6 and tumor necrosis factor alpha, thereby ultimately reduces the nociceptor or pain sensitization. The next one is short-chain fatty acid producing bacterium. These are Faecalibacterium grudnense, Roseburia intestinalis, Bacteroides and Lactobacillus. All these bacteria will produce a metabolite which is known as butyrate and will also produce acetate. The main role of butyrate is to inhibit the histone deacetylase and decreases the neuroinflammation. This butyrate can also modulate the microbial activation in the spinal cord. As I told before, it modulates the microbial activation and thereby enhances the endogenous analgesic pathway. This reduces the requirement of intraoperative opioids. The third is Bifidobacterium longum. This also has its influence on gut-brain axis by acting via the vagal pathways. It increases the GABA production, which is an inhibitory neurotransmitter, and enhances the serotonin precursors, which are tryptophan. These are pain modulators. Thereby it improves the pain tolerance. What is a preoperative strategy? The main strategy is to create a microbiome conditioning protocol. This has two components, which is probiotic supplementation and prebiotic diet. This probiotic supplementation has to be started 1 to 2 weeks before the surgery and started the beneficial bacteria. As I told, the prebiotic diet includes high fiber diet. What is the role of prebiotic diet? It is to promote the short chain fatty acid production, ultimately improves the gut barrier function. What are the expected benefits? It reduces the inflammation and the perception of the pain is reduced or lowered and the opioid requirement is decreased. All these will ultimately lead to better recovery outcomes. So what are the advantages of this technique is that it is a non-invasive intervention, it doesn't include any injections and it can be easily implemented. Instead of suppressing the anesthesia practice, it complements the anesthetic practice. There is less use of dangerous anesthesia drugs. This requires more number of clinical trials to validate our concept and the doses and duration has to be optimized. Microbiome profiling has to be done. That is, we have to find what kind of bacteria is in the gut and in which quantity. And this approach has to be integrated into ERAS protocol, that is early recovery after surgery protocols. I'd like to tell it in a short way what my idea is. This approach focuses on preconditioning the patient microbiome prior to surgery. How this works is, it is based on gut-brain axis, which is a communication between the gut and the brain. When we improve the composition of the gut, we can do the following things, which I will be telling now, which is decreasing the systemic inflammation, decreases the nociceptor sensitization, that is pain sensation can be decreased and modulates the central pain pathway. There are two components, which is the probiotic supplementation and prebiotic diet. The probiotic supplementations are Akkermansia, Bifidobacterium, and prebiotic diet includes high fiber diet. This high fiber diet will produce a strong anti-inflammatory and analgesic effect. These two, the Akkermansia and Bifidobacterium, are also available as a capsule in a powdered form. This can be administered to the patient 1 to 2 weeks before the surgery. In conclusion, I like to tell microbiome modulation is promising and is basically targets the gut-brain axis, transforms the pain management. This is a conceptual hypothesis which requires further validation and research before coming into regular or routine practice. Thank you.
Is it my voice is clear?
Yes, sir.
Okay. The excellent presentation. The slide number and the content of the presentation summary would have been better. Otherwise, it's fine. It's a very important area as point of doctoral student. I would like to ask you. Doctor is going to administrate antibiotics before surgery. So is it fine? Because gut bacteria, antibiotics and gut bacteria can go together.
Yes, sir, they can administrate. That's why I've given a time period of 1 to 2 weeks before the surgery. As you know that the generation time of bacteria is only 24 hours, but there are many bacteria in our gut. So to stabilize our gut, we are giving a time period of 1 to 2 hours, 1 to 2 weeks before the surgery.
Okay. What is your general suggestion for a human being to maintain the gut bacteria in real life?
In real life, I would suggest them to take a high fiber diet, like they can take vegetables, and also they can take fermented food items, which are rich in lactobacillus, curd, milk, and the dosa batter is also good for health. So I suggest them to take all these.
You are suggesting for fermented foods better than normal fermented probiotics, like probiotics.
Sir, probiotics as well as the diet has to be should be taken together, sir, for better effect. So why I am telling this is because when we are giving a prebiotic influence, that is when we are giving a high fiber diet, it will cause the growth of the friendly bacteria in our gut, which are excluded the pathogenic growth of other bacteria. These friendly bacteria will do is they will release the postbiotic, which will help to maintain the homeostasis of our body. That's why I'm asking, and I'm having an idea to take together both prebiotic as well as the probiotics.
Okay. Then as well as I have passed the article recently. The gut bacteria is really giving trouble to the brain. Exactly. Is it what way is disturbing the brain, the brain activities particularly? We are simply thinking that the bacteria will be working in the only gut.
So your voice is not clear.
Yeah, the gut bacteria, how is giving troubling to the brain cells?
They will competitively exclude the pathogenic bacteria, sir. That's how they work, and they will allow the friendly bacteria to grow.
Okay, ma'am. Thank you, ma'am. Bye.
Thank you, sir.
Oral medicine. Post-end.
We will now transition to our online poster presentation session. Now I invite our first poster presenter, Divya Darshini, to share their screen.
இது வந்து ஓரல் போஸ்ட் பண்ணி
Divyadarshini, can you hear me?
சார் நெக்ஸ்ட் போயிட்டு காமிச்சிட்டு விளாண்டுவோம் எங்கள் பசங்க மேம் சொல்ல பார்த்தீங்க போகலாமா
இங்கே இருக்கு பேகு இங்கே இருக்கட்டும் இங்கே ஃப்ரெண்டில் வச்சுட்டு
Okay, then I will now invite our first poster presenter, Sandhya, to share their screen.
Okay.
Sandhya, can you hear me? Good morning, everyone. I'm Divyadarshini, a second year biotechnology student from Srimati Indira Gandhi College. I'm going to present my reference-based research title, Production of Biology of Environment IPA. Divyadarshini, your screen is not clear. Simon, your screen is not clear.
Charge ಎಲ್ಲ
Can I continue?
Can you please auto rotate your phone? சொல்லு யோகா
You can auto rotate your phone, please. How will I calculate?
அவளை அடுத்து இது பண்ணிக்கோங்க நெக்ஸ்ட் பர்சன் போகணும்
விஜயதசமி வெயிட் பண்ண
Divyadarshini, can please wait. Now, I would like to call upon Ranjani to share their screen. Ranjani, can you hear me? Vinita, are you there? Ranjani, are you ready to present?
ஃபோன் இருக்கா
அவங்கள வெயிட் பண்ணுங்க. அந்த லிஸ்ட் கொடுமா.
நான் தான் பேசிட்டு
போட்டு அந்த பர்சன் ரஞ்சனி கூப்பிடுங்க.
Ranjani, please start your presentation. We share our screen.
இதோ கால் சென்ட் பண்ணுங்க.
Good afternoon, everyone. Myself, Section of the Bachelor Engineering College. Today I am going to present my course on child learning knowledge, learning sustainable agriculture and environmental conservation. Ranjani, your voice is not clear. Technology means plant and microorganisms to improve agriculture in an eco-friendly way, and it is also focused on increasing crop production and protecting the environment. Ranjani, your voice is not clear, please. In my continue, natural biological method in farming. It helps farmers grow healthy crops and reduce pollution.
Ranjani,
Yes, ma'am.
your voice is not clear. Ranjani, are you ready?
Yes, ma'am.
Okay, now you can present. Ranjani, please start your presentation.
Good morning, everyone. Myself, Ranjani, second year technology, Engineering College. Today I am going to present my course on Green Life Technology, Advancing Sustainable Agriculture and Environmental Conservation. Green Life Technology means plant and microorganisms to improve agriculture and an eco-friendly way, and it is also focused on increasing crop production. To begin the introduction, Green Biotechnology use natural biological methods in farming. It helps farmers grow healthy crop and reduce pollution. The main objective of this study to increase crop production, to improve soil fertility and promote eco-friendly farming. Moving on to the methodology. Use the genetically improved crops. Use the bioremediation to clean polluted soil and water, followed by organic farming methods. Stop that result, crop production increase, pollution reduce, soil quality improve. Use less of chemical farming for better ecosystem stability. In conclusion, green biotechnology is safe and eco-friendly method and support agriculture products, nature and health achieve sustainable development. Thank you for this opportunity.
Ranjani, can you hear me? Ranjani?
Yes, sir.
Whether your poster is good, but whether you did any field work for this, the title also is good. But it is a kind of the review paper you are presenting. Hello.
Yes, sir.
Whether you did any field work for the sustainable agriculture.
Yes, sir.
The agriculture நீங்க ஏதாவது field work பண்ணீங்களாமா இந்த டைட்டில் வச்சுட்டு?
Sir, your voice is not audible, sir.
இந்த அக்ரிகல்ச்சர் அப்ளிகேஷன் தான் கொண்டு போறீங்க இல்லையா? என்விரான்மெண்டல் கன்சர்வேஷன் அண்ட் அக்ரிகல்ச்சர் தான் கொண்டு போறீங்க, சஸ்டைனபிள் அக்ரிகல்ச்சர்.
Yes, sir. இந்த மெத்தட் நம்ம யூஸ் பண்ணும்போது அக்ரிகல்ச்சர் சாயில்
ஏதாவது ஃபீல்டு ஒர்க் ஏதாவது பண்ணிருக்கீங்களாமா? கேஸ் ஸ்டடிஸ் மாதிரி ஃபீல்டு ஒர்க் ஏதாவது பண்ணிருக்கீங்களா?
பிறகு பண்ணிட்டு இருக்கேன்.
இதெல்லாம் பண்ணிருக்கீங்க. ஓகேம்மா, குட்மா, ஆல் தி பெஸ்ட், பை.
I now request our next participant, Niveta, to share your screen.
ಯಾರು ಮಾಡೋದು? ನಿವೇತಾ.
Niveta, are you ready? Niveta. Yes, ma'am. You can start now.
ಅವ್ರ ಸ್ಲೈಡ್ ಇರ್ತದಮ್ಮ.
Niveta, can you please share your slides? Are you there? Niveta, we shared your poster, please. You can start now. Sandhya, are you ready, Sandhya? Sandhya. Sandhya. Sandhya, can you please share your screen? Can you please share your screen?
வாய்ஸ் பிரேக் ஆகுதா, கிளாரிட்டி இருக்கான்னு கேள்வி. கொஞ்சம் அவங்களோட ஆடியோ ப்ராப்பராக வச்சுக்க சொல்லுங்க.
Niveda, are you going to present or not? Now you are sharing your screen. Can you please talk, Niveda? Okay, Sandhya, can you please share? Okay, ma'am.
Good morning, everyone. Myself, M. Sandhya, second PhD Biotechnology from Srimati Indira Gandhi College. Today I am presenting my poster on anti-cancer potential of marine natural product, mechanism and application and clinical advances. To begin with,
Sandhya, please wait, we will share your screen. Your poster is not clear. We will share your poster.
I can share it now. I'm already sharing it.
Yes, we, Sandhya,
Good morning, everyone. Myself, M. Sandhya, second PhD Biotechnology. Today I am presenting my poster on anti-cancer potential of marine natural product, mechanism application and clinical advances. To begin with that, the topic focuses on how natural substances are derived from marine organisms like algae, sponges, and microorganisms can be used in a cancer treatment. To begin with, the marine organisms contain unique bioactive compounds that share the strong anti-cancer properties. This compound kill cancer cells, inhibit their growth, and prevent tumor formation. On important advances in this, they have fewer side effects compared to conventional drugs. The main objective of this study is to analyze this marine derived components under the mechanism of action against cancer and evaluate their potential in medicine. Moving on to the methodology, samples were collected from different sources. The bioactive components were extracted and isolated and then tested on cancer cells under laboratory conditions. As the research showed that this components have significant anti-cancer activity, they were effective in introducing cancer cell death, reducing tumor growth, and showed lower toxicity when compared to existing treatments. Conclusion: marine natural products are anti-cancer agents. They are safe, effective, and have great potential in the development future. Thanks for valuable time. Thank you, sir.
Sandhya, can you hear me?
Sir, I was audio.
Can you hear me? Hello? Hello, Sandhya?
Sir,
Hello?
I can't hear, sir.
Yeah, can you hear me?
Yes, sir.
So what type of marine natural products you are proposing? You didn't mention any one of the products. or what type of cancer you are also focusing. But those things is missing in your poster. Okay. Yeah, try to include those things when you go for the next meeting, next conference. Okay. But you are BSc or MSc? Hello. Hello. Sir. Can you hear me, Sandhya? Hello.
Sandhya. Sandhya. Sandhya.
Thank you, ma'am. Thank you, Sandhya. Bye.
Next, I would like to call upon Niveka to present. Yes, ma'am. Wait, we will share your poster. Then you can start. Your voice is not audible, ma'am.
நம்ம ஆடி வர்றாரு
Niveka, can you audible? Yes, ma'am. Is it audible? Is it clear? Yes, ma'am. Yes, you can share your presentation now. Ma'am, can you share now? You can start your presentation. Is it visible? Can you see the poster? Yes, ma'am. You can start. Yes, you can start. Good morning, ma'am. Good afternoon, judges. I am being Niveka from second BSc Biotechnology in Srimati Indira Gandhi College. Today I am presenting my reference study result AI unlock English microbes for new medicines. The core problem we are addressing is that our 99% of marine microorganisms cannot be grown in the laboratory. They are unculturable. However, these deep sea microbes will extreme environment and produce unique chemical compounds that could be next generation of antibiotics or anti-cancer drugs. How to solve the problem not being able to grow these microbes, we use two powerful tools, metagenomics and artificial intelligence. Step 1. We collect the water or sediments from their Step 2. We extract environmental DNA directly from the samples because isolated individual species. Step 3. We perform metagenetic sequences to read the genetic code. Step 4. We create a massive amount of data. Then we use AI algorithm to scan this sequences. Step 5. So AI will identify biosynthetic gene clusters, BGCS. These are specific group of genes responsible for producing additional compounds. The impact. Studies shows this approach is significantly faster and more efficient than the traditional methods. By using AI to predict which genes will produce useful bioactive compounds, we can skip the year of trial and the error usually required to drug discovery. In conclusion, the combination of metagenomics AI allows us to tap into the new chemical structures of the oceans.
way of right-feeling issues in this future potential threat under concern.
Have you completed? Niveta. Yes, actually your title is good. That is in the resulting math.
Yours is not clear now.
Now it is clear? Now?
Yes, ma'am.
Yes. Actually your topic is very nice. Are you a MSc student or a UG student or PG student?
Actually, ma'am,
PG or UG student?
PG student.
PG student. You are in first year or second year?
Second year, ma'am.
Second year. Have you started this work? Have you started this work?
Actually, ma'am, not clear you are asking.
Have you started your researcher, this researcher?
Now we have start my research.
Can you mention any two of your objective of this study? Objective of the study. Actually, I explained the abstract, the methodology, everything here now. So, can you explain any two of your objective of this research?
Yes, ma'am, I will tell you.
Yes.
Good afternoon, ma'am. Good afternoon, ma'am.
Good afternoon. What is your first objective of this work?
Ma'am, your voice is not clear, ma'am. Your voice is not clear, ma'am. Once again, ask me a question, ma'am.
Where did you doing this research? In your laboratory or outside of your institution?
It's my lab, ma'am.
Huh?
It's our lab, ma'am.
Niveta,
Yes, sir.
your title is very attractive. That is why we are asking more questions. So, what are the ocean microbes?
Sir,
You ask me ocean microbes. Yeah, ocean microbes are also young. So, for the medicinal application, so what do you want?
Yes, sir. Once again, ask me a question. Yes, sir.
Okay.
சரி ரிசர்ச் ஸ்டார்ட் பண்ணிட்டீங்களா இல்லையாம்மா?
Yes, ma'am. இப்பதான் கிளம்புறேன்.
பண்ணிட்டீங்க சரியா? இதுல ஆக்சுவலா ஒன்று ஒர்க்கு கிடையாது. பட் ரெஃபரன்ஸ் எடுத்துக்கணுமா இல்லையா? மெத்தடாலஜி நீங்க இன்னொரு பேப்பரை ரெஃபர் பண்ணிக்கீங்க அப்படிதானே? ஓகேம்மா, நைஸ்மா. தேங்க்யூ.
Thank you for this wonderful opportunity given me.
ஓகே.
Thank you.
திவ்யாவா?
Now I would like to call Dani to present.
போஸ்டர் நீங்க எல்லாம் மொபைல்ல வச்சிருக்காங்க யார்?
Dani, we can share your poster. You can start the session.
Yes, ma'am.
Please wait. We can share your poster now.
Ma'am, can you please share my post?
Wait, now we will share.
Ma'am?
சாட்ஜிபிடி ஃபார்மேட் தானே? சாட்ஜிபிடி ஃபார்மேட் அப்படியே எடுத்துருக்காங்க டாபிக்.
No. I'm talking here. Not about you.
No, I edited now.
Yeah, it feels good now. No issue.
Dharani, please start your presentation. Okay, madam. Good afternoon, everyone. Myself, R. Dharani, from 2nd BSc Biotechnology. Today, I am going to present you my poster on microbes mantle screen role in bioremediation and environmental sustainability. To begin with, microorganisms like bacteria and fungi and to help to clean the environment. This process is called bioremediation. And they are naturally found in soil and water and helps to maintain ecosystems. The main objective of study is to understand how microbes remove pollutants in environment setting. Move on the methodology. In this method, we use microbes such as pseudomonas, bacteria, and aspergillus. These microbes produce enzymes that break down pollutants. They also remove heavy metals from contaminated areas. Finally, as a result, pollution in oil spills, wastewater, and soil is reduced. The environment becomes cleaner and capacity levels. So microbes are very useful. They provide a low cost and eco-friendly way to control pollution and protect our environment. Finally, I conclude the microbes play an important role in environmental sustainability. Thank you.
Darling? Hello?
Yes, sir.
Are you studying BSc or MSc?
BSc, sir.
BSc third year.
Second year, sir.
Second year. Yeah, but your slide, the topics you focused, but it is very, very important topic. Whether you conducted any studies in your laboratory?
No, sir.
No. Whether you conducted any studies, bioremediation studies in your lab?
Sir, your voice is not clear, sir. No, sir.
Darmi?
Yes, sir.
ஏதாவது ஸ்டடிஸ் இந்த லேப்ல பண்ணிருக்கீங்க, அதுக்கான ஸ்டடிஸ் ஏதாவது சூடோமோனாஸ் பேசிலஸ் இதெல்லாம் ஒத்துழைக்கீங்க இல்லையா?
No, sir.
நோ சார் இருக்கீங்களா? எஸ் சார் இருக்கீங்களான்னு புரியல. நோ சார். Then how do you define the methodology and result and conclusion without doing anything?
Sir, bioremediation பத்தி நான் எதுவும் பண்ணல, sir.
இல்ல, ரிவ்யூ தான். இது ரிவ்யூ போஸ்டர், கரெக்ட்?
Yes, sir.
என்ன பண்ண, since you are the BSc second year, you have some more information, even though it is a review, but you try to do some work, join with your MSc students or and then do some lab work as well. Otherwise, you will lose some images here and there. Okay, great, man. All the best. Bye.
Thank you, sir.
Bye.
Divulgation, are you ready to present? Divulgation, are you ready to present? Yes, sir. We will share your poster. You can start. we start now? We can start now. Good afternoon, everyone. I am Divya, a second year biotechnology student from Srimani Indira Gandhi College. Now I am going to present my reference-based poster on production of biofuels from live algae. This reference study focuses on live algae as a third-generation biofuel resource based on previous research findings. Live algae show how lipids productivity, rapid growth rate, and ability to grow in seawater without competing with agricultural land freshwater resources. The objective of this reference study is to analyze existing research findings on marine algae and evaluate their potential role in renewable biofuel production. Starting with marine algae, they are photosynthetic aquatic organisms capable of converting sunlight and carbon dioxide into energy-rich compounds according to several research papers and reports. Species such as Rhamnochloropsis and Chlorella are widely studied for their biofuel production potential. The process begins with cultivating marine algae, when the algae harvested and oil is extracted to produce biofuel. The remaining biomass is further used to produce bioethanol and biogas, ensuring efficiency energy utilization. Based on the analysis studies, marine algae show high biomass productivity, strong lipid content, and environmental sustainability advantages, making them one of the most promising renewable biofuel resources. So through this reference study, I conclude that marine algae are not just studied as an amazing energy source, but recognized globally as the future directions of sustainable biofuel resources. Thank you.
Divya? What are the marine algae? Whether you conducted any studies in your lab work?
Sir, no, sir, I didn't conduct any experiments. I collected the information from review articles, sir.
Okay, just this is kind of the review question, right? The person of review was not complete. It is better to research paper. Yeah, paper question, research paper, journals. Hello, research paper, the post is there.
Sir, I can't hear you clearly.
Okay, bye.
Divya, my first
Now I invite Mohammed Harris to the podium for the oral presentation. I will Mohammed Arif, you can start your presentation.
Good afternoon, everyone. I am Arun.
Yeah, your time is 3 minutes.
Okay. I am Arun Karan and I am along with my friend Mohammed Arif. We are from the Biomedical Department, ED School Engineering College, Nagapattinam. Today we are presenting our work, eco-friendly synthesis of copper oxide nanoparticles from Lucas aspera, extract for faster antimicrobial wound healing. And then abstract. Let me quickly summarize the overview of this our work. The main problem is cotton coating in hospital do not have any antibacterial activity. So we address this problem to develop a synthesis of copper oxide nanoparticle using Lucas aspera plant. So in Tamil it's called Tuppaichiri. And then the synthesis is using a green synthesis method and then coated this nanoparticle into cotton using dip coating technique, and then you confirm characteristic study is like UV visible spectroscopy and then SEM imaging and then in the result we confirm the strong antibacterial activity up to 28 millimeter inhibition. So there is so basically it is eco-friendly, effective and practical. Now coming to introduction, wound infection is a serious problem because it will delay wound healing and can cause infection.
Your materials and methods, that means methodology.
Okay. So problems will be
மெத்தடாலஜி போகுமா என்ன பண்ணீங்க?
Good afternoon to all. Myself Mohammed Arif. First of all, our objectives is main objectives eco-friendly material to extract copper oxide nanoparticle from the copper salt, so that we can using a copper salt copper oxide nanoparticle to enhance the immunity so that inhibit the growth of the bacteria in the wound. So that integrating the copper oxide nanoparticle in the cotton is the first objective. Next slide, bro. This is a wild Lucas aspera. As we see in Tamil, it is called as Tumbai. Lucas aspera is here. Enhance the rich phytochemical content of alkaloid, so that it can enhance the wound healing. So that we selected Lucas aspera as a reducing agent and substitution agent. So that it is a cost effective and easily available in the countryside. Then tea and neem, turmeric. This is also possesses strong antimicrobial activity, but that's not suitable for the sustained release and release of nanoparticles. And also if we extract a copper oxide nanoparticle with it neem or turmeric mix, the aggregation of the nanoparticle may be occurred. Next slide. This is the literature view. We can refer the three papers so that we can find limitations. First paper we can found a limitation that is
Move to methodology. What you did?
first of all, plant selection. That the plant is a Lucas aspera. We can take a 100 gram, 25 gram of Lucas aspera in a lab scale. We can take a 25 gram of Lucas aspera, and then the leaves are dried and the that is added in the 100 milligram of water. So that after 100 milligram of water, next slide. 100 milligram of water and is heated and filtered is a Wartman river. We can make a lake solution that can contain a Lucas aspera extract. Next slide. Then we wanted to make a copper oxide nanoparticle so that we can add a filtrate, the Lucas aspera filtrate with the drop wise in the copper sulfate solution. How we make a copper sulfate solution means CuSO4, that is a chemical formula of copper sulfate. We can add 0.5 gram of copper salt in 100 ml of water, and we add a filtrate in a drop wise, and when we adding the filtrate in a copper sulfate solution, the temperature maintained in 37 degrees Celsius. And then drought wise, we can add a lead solution into copper sulfate solution. The blue color is converted into a dark brown color that indicates the copper acid nanoparticle formation. Next slide. So that then we made a copper acid nanoparticles, so that we want to embed the copper acid nanoparticle in the cotton, so that we can achieve the enhanced wound healing property. So that we use a dip coating method, just to dip the cotton in a solution, so that the copper acid nanoparticles are embedded in the cotton. Next slide. This is the resultant discussion. We can first scanning electron microscopy to see the copper oxide are formed or not, so that it can clearly be visualized. The particle like you said, slightly aggregation. Then rough surface of a nanoparticle indicates the high efficiency. Then the X-ray diffraction. This peak shows the crystal energy, high crystal energy, so that this is a pure nanoparticle, indicates the pure nanoparticle formation. Then copper oxide nanoparticle size from the 20 to 100 nanometers. This is the nanoparticle is spherical in shape. Next slide. And then this is the UV visible absorption spectroscopy. This also used to indicate the formation of the copper oxide nanoparticle. This peak between the 380 nanometer to 420 nanometer. This indicates the formation of the copper oxide nanoparticle. Next. Then it's an antibacterial studies. We can collect Staphylococcus E. coli, Corynebacterium, micrococcus like bacteria from the microbiology lab, so that using this bacteria with our research, we can achieve this time of inhibition. It is measured in the millimeter. We can use the other plate for the.
Can you show the plate?
Other plate.
Can you show the plate? You have?
Sir, I did not include that plate, sir.
How come it's possibly at 28 mm, 28 mm, 27 mm? That is what you have to, when you want to prove this data, you have to show the plate. Otherwise,
You can use any number. Okay? It is maybe imaginary number.
Sir, you have a manuscript also, sir.
No, you have to for the presentation.
Have you
You have
published
to.
there? Have you
Yes,
published?
ma'am, completed. We have a ticket.
Have you published? Published?
Still, these are published, ma'am. Our mentor,
No, actually, that is authentic research, scientific research.
Yes,
We need
ma'am.
to project for this. The table is not sufficient to confirm the.
we plan to publish the paper.
even though you have the
Good data, then you will come along with the image. That is necessary.
So this is the conclusion. Using the copper oxide nanoparticle, we can achieve the enhanced wound healing, so that leukous aspirate act as a stabilizing agent, and also leukous aspirate helps to increase the immunity, inhibit the bacterial growth. As we saw in diabetic patients, leg may be injured, the less immunity can cause a prolonged injury. Okay. So that we can use embedded in the copper oxide nanoparticle embedded in the socks means we can inhibit the growth of bacteria, then also we can increase the immunity. So that this is a plan embed the copper oxide nanoparticle in cotton to increase the immunity, so that we can achieve faster healing and enhanced wound healing. Then it will be a cost-effective one and eco-friendly one.
okay, fine.
So then you mentioned your nanoparticle formation at 37 degrees centigrade. Whether it's possible to form the nanoparticle at room temperature?
No, sir.
Then you said 37 degrees centigrade.
So 80 to 90 degree Celsius, sir.
And then when you discussed, you said 37 degrees centigrade. That is what. But what is the size of your nanoparticle?
Size may be, sir, 20 to 100 nanometer range, sir. But in lab scale, the eco-friendly method, so that size may be very small. So that 20 to 40 nanometer size.
You have to protect those things. Your conversation you are telling, but you are not at all showing the size
and the
Concentrate
confirmation.
on your image, what you have given on that. Can you mention the type of mechanism that is involved in antimicrobial activity? How it can produce the antimicrobial activity? One and only question.
It is reactive oxygen species.
Reactive oxygen species.
Copper-oxide nanoparticle release.
How it can control the microbial growth? Just explain in one line. One line.
Reactive oxygen species. It is copper-oxide nanoparticle release the copper Cu2 plus ions. So that it reacts with the oxygen to form a reactive oxygen species. It kills the cell membrane and destroys the bacteria. As we also listen to the nanoparticle, ma'am.
If it is affected the cell membrane, how it can be degraded? How the microbe cannot grow? How the microbes cannot grow after that? No, just all the
கான்டாக்ட் வச்சிட்டு இருக்கிறப்போ அது மறுபடியும் க்ரோ ஆகாது மேம். இன்ஹிபிட் பண்ணிடலாம் மேம். It can
how it can inhibit? How it can inhibit?
It sustainably release the copper oxide
How
nanoparticle.
it can inhibit after destruction of the cell wall? That's my question.
It destroys the reactive oxygen species, which is destroys the cell wall.
Okay,
It destroys the
all
bacteria.
the best. Next person.
Also on this silver, they use the silver, but silver is a rapidly releases the ions. So rapidly releases the ions. So on there,
Yeah, your work is so good and your explanation is also good. Wish you all the best.
normal humans எல்லாம் வந்து destroy பண்ணிடுறாங்க. But copper oxide nanoparticle sustainably release the ions. So வந்து sustainable release பண்றப்போ அது வந்து human cells damage பண்ணாது.
Okay, good.
Reactive oxygen species மூலமா அந்த மெக்கானிசம் மூலமா அது destroy ஆகிடும்.
Okay, good.
Thank you.
Thank you for your presentation. Now, next participant, Sri Nandana and Kausalya. Note that you have 3 minutes for your oral presentation. Please finish that.
Ma, your time is only 3 minutes. Try to complete it.
Good afternoon, everyone. I am Sri Nandana and this is my teammate and Kausalya. We are pursuing a second BSc Biotechnology in Srimati Indira Gandhi College. Today we are going to present a modular nanotechnology integrated with the RFID system. In modular nanotechnology integrated with the AM frequency identification system, enhance the traditional RFID system with a minimization, sensitivity and multi-functionality. Integration uses nanomaterials like carbon and graphene nanotubes which are create ultra small and durable, packed with enhanced data storage and signal transmission. Our key applications include cancer therapy or cell control nanoparticles hyperthermia. In a hyperthermia, the heater to kill the cancer cells and supply the chain management and environmental sensing and challenges like high cost and safety concerns. Molecular nanotechnology focuses on designing and controlling materials like the molecular and atomic level to create a highly efficient nanoscale systems. When combined with the RFID system, it improves the tracking, sensing and communication, leading to a smart and miniaturized system used in healthcare and environmental monitoring and industry. In objective to understand the basics of molecular nanotechnology and RFID system in integration of nanosensors with the RFID technology to improve minimization and efficiency of RFID device use the nanotechnology to analyze the application in healthcare and tracking and security systems and challenges and future advancement in MST on RFID system indicator. Molecular nanotechnology. In molecular nanotechnology works at the molecular and atomic level. Nanoscale size is 1 to 100 nm. Build a material using a molecule, create a nanodevice and nanosensor used for medicine and electronics. In RFID system, wireless identification technology use the radio waves, hashtag reader and antenna and track for identifying objects. And the type passive RFID is a sensor and active RFID is a nanodevice. Semi-passive RFID system, nano biosensor, environmental sensor, RFID and smart nanosensor networks. Nano electronics fast low power of property. Nano applications that create small RFT tags, graphene and carbon nanotubes, biosensor and nanosensor, wireless communication IoT. In biosensor and nanosensor, delicate byte and chemical signal, wireless communication, data transfer, IoT for smart monitoring. Application: cancer treatment, cellular system, patient health monitoring, smart biosensor for challenge, high prevention cost, technical complexity, safety control, data privacy, difficulty in regulatory approval, future development, advanced smart nanosystems.
Okay, ma'am. Stop it. Thank you. What is your contribution in RFID, application of RFID? What is your contribution? These are the review. Okay. What are you thinking? Since you are a biotechnology or ECE student?
Biotechnology.
Both are you biotechnology?
Yes, sir.
Only one objective from you. Whatever the things which is not existing so far, I am planning for this. Like this, you have only one idea, just tell me, then close it.
Sir, in AI and IoT did not consider in advanced one because the cancer in hypothermia to treat the body tissues and the heater to the cancer cell, it's a helpful for RFID system, sir. So we use it.
Mean that can diagnosed by RFID, isn't it?
Yes,
Helpful for hypothermia treatment.
Yes, ma'am.
Hypothermia is a very old treatment. Actually, decades before actually the technology was started.
Yes, ma'am. It's already old method, no?
What is the innovation of this study?
Sir, this is the AI and IoT, we are going to project this topic.
What you did, that is what the sir asked. Have you started this one?
No, ma'am.
So no more contribution on this
We are
one.
going to.
Okay, good. All the best.
Thank you. Thank you for the presentation. We will now proceed to the next participation. Now I invite Nikita and Madhumita to present.
Good afternoon, ma'am. Good afternoon, sir. I am Nikita, joined to my teammate Madhumita, representing from Srimati Indira Gandhi College. We are here to present our topic, AI-driven drug discovery, transforming modern medicine. By integrating deep machine learning, deep learning, and data analysis, AI enables scientists to discover the drug. Introduction: drug discovery is a time-consuming process which takes 10 to 15 years and a quite expensive process. Average cost exceeds 2.6 per drug. AI enables faster, accurate, and cost-effective drug development program. Why we have used AI in drug discovery means a traditional method relies on trial and error system. Need for a rapid drug development is used. What is AI in drug discovery? We have used this machine learning, deep learning, and neural network and data analysis system in drug discovery. It helps in targeting, identification, drug design toxicity, and prediction. Our workflow: first, data collection. This is our workflow: data collection from genomics and proteomics. Next, target identification, ritual screening. Then it leads to the optimization and preclinical and clinical. These are the existing work. This is our review paper. The existing work which focuses on individual states, but we are going to innovate through covers complete drug discovery pipeline, uses separate AI technique and integrated approach, focuses on practical healthcare application and outcomes. Our project mainly focuses on these applications: drug target identification, repurposing the predicting drug, and toxicity personalized medicine.
It reduces time from 10 to 15 years around 5 to 7 years. It costs cost reduction up to 40 to 60%.
AI have high accuracy prediction, handles large data sets issues. Its limitations, AI requires high quality data, expensive computers and technologies. AI is ethical in data privacy concerns. We have sometimes we have a black box problem. Clinical trials consume the majority of drug development. Compared to traditional method, AI reduces the drug discovery. Future advancement.
Okay, ma'am. Great. What is from this title? What is both of you? What you can contribute? So far you have designed with the existing things. Wherever you can see, you can get this kind of information. From this, what you come out, what you are trying to say, what is your contribution is supposed to be in the future. Since you are the biotechnology.
Yes, sir. In traditional so far the methods followed, we have followed the traditional methods, sir. It takes 10 to 15 years. We have contributed in the AI, now current growing technology field, sir. Using the AI, we can use most precision result we can give, sir. Precision result, faster and accurate result we can give using it in practice.
Yeah, it's good. This is also general statement. Precisely which issue you can target by minimizing the time, minimizing the expenses in the laboratory. What way you can do. So that way you have to come out.
Yes.
But that kind of idea is not existing right now.
Yes, sir. Yes, sir.
Okay, all the best.
Thank you, sir.
Thank you for the presentation. Now I would like to call upon Pratibha and Kanuri to participate.